Hey, everybody. Please post your questions in the chat. I'm going to, apparently the link to the email list form is not working. I am going to try and jump on that very quickly, but feel free to post questions in the chat. And Brian, if you could please read those questions, I'll get on this real quick.
Sure. Our first question is, does this detect longfin smelt?
Joe or Susie, can you address that? I'm not sure if that's in the library.
I think we just checked that one. Nick, give me a thumbs up. I think longfin smelt. Well, so in general, for all intents and purposes, all fish will be detected with a process like this. The question is whether first that specific species is in our library, so whether that's been sequenced yet, and then whether that species is going to be sufficiently resolved so it has differences in the sequences such that we can resolve that species from other species. And I think in this case, longfin smelt is in the library and can be well resolved. We have a list, I'm not quite sure where it is, I have to dig it up, of what species known to be in California have already been sequenced and which ones can or cannot be resolved at this point. So that's something that we will get out to everybody. And probably part of this process too will be to start to identify the species that really need to be sequenced soon and figuring out ways to do that.
Okay, thanks, Joe.
Mm-hmm.
Our second question here is, how are you working with abundance data? Is it being normalized against a mock community, or is there another method you're using?
That's a good question too. So the data that we get back, so there's a series of steps that happen that Peter began to outline. So the first thing we do is we extract the DNA that's on the filter, and then that DNA using primers, which are short, relatively conserved regions in the chloroplast or on the mitochondria, for example, will be used to then make lots of copies of the DNA so that we can eventually sequence it. When there's a lot of DNA on a filter, like with phytoplankton, then the relative number of reads that we get back, so how many sequences we get back associated with a given species for a sample, is relatively proportional to what was there initially. And so therefore, if we get 50%, 25%, 25% for 3 different taxa, then we can make a pretty good assumption that it was 50, 25, 25, so 2 to 1 to 1 initially. For something like fish, as we get to the other end of the spectrum, that their DNA is relatively rare in the environment and relatively rare on our filter, such that when all of the PCR occurs, those original proportions aren't necessarily kept. And so for something like fish, we say that it might be semi-quantitative, but in general, when we use this approach of metabarcoding, we tend to, for a particular sample, just think of that as a presence, as opposed to actually a relative abundance, whether that's the number of individuals or the biomass or activity, anything like that. Other techniques potentially can be used to get at abundance, but those are kind of tricky. So as far as fish are concerned, we tend to think of that as just presence. That people might ask questions about bioassessment and how we do our indices and how they're going to change when we start to go to eDNA as opposed to using more traditional techniques. We can talk about that later if somebody thinks that's a good question. Levi, does that answer your question pretty well?
Yeah, it does. Thank you very much.
You're welcome. Thank you.
And the next question here is, could the list of species in the library be made available?
Yeah, really easily. I know that we have a file, there's somewhere on my hard drive. I can send that along and that'll show you which ones have already been sequenced and which ones need to be. And then hopefully we can figure out how to get DNA for all the ones that have not been sequenced yet, so we can plug those holes pretty quick.
And with that, we're caught up with the questions in the chat.
Why don't you tell us about how metabarcoding for macroinvertebrates can help, please, Joe?
can help.
Sorry, how that process is going.
Can you give me more to go on just how it can help? Right, so so the phytoplankton side, sorry, for each taxonomic group, we have different bottlenecks in really getting this to be effective. For fish, for example, we've had to work on detection a little bit and spend some time on that to make sure that when we get a sample, we're getting something that we can think of as being representative of what's actually in the assemblage, and then trying to figure out how many samples we have to take for a particular stream or river in order to well characterize that one. I can talk a little bit about that later too. On phytoplankton on the other side, then for all intents and purposes, the sampling is really easy. It's the libraries and the identification of the sequences that require a fair amount of work. So out of the thousands and thousands of phytoplankton species that would be there, really maybe 500 have been sequenced, something like that. So we haven't really caught up to traditional techniques necessarily as far as identification is concerned, and that's going to take a major push. Macroinvertebrates is somewhere in the middle, where we've got some new challenges there too. So first off, when people have done bioassessment and indices for macroinvertebrates, that's essentially empirically everything that ends up in a kick net, right? So it's everything that doesn't have a backbone that you can kick and get to go into a net. And so in that case, they're actually phylogenetically, evolutionarily really different. So you have small clams on one side versus insects, and even insects is a relatively diverse taxonomic group. And so to find one conserved region in its that only will amplify the DNA of the ones that we care about, for example, just of the insects and maybe some of the mussels, that's relatively difficult. And even just for insects, that's hard too. So we've put in a fair amount of effort, as have other people in labs, to try and figure out how to get which DNA sequences to go after. And we're still kind of working on that. So the data that will come out this year is definitely preliminary, and we're hoping to get that to be improved. In addition, our libraries aren't complete for insects, that's for sure too. So even though there's been a heck of a lot of work for people sending individual identified insects to DNA repositories to be sequenced, there's still a lot more that has to be done. So in all of these cases, it's going to be iterative. The sequence that we find on a filter that we sequence, that's immutable. That isn't going to change. All that's going to change is who we associate, which species we associate that sequence with. So that's going to take a fair amount of work. If there are any taxonomists that were a little bit worried about job security with the revolution that's associated with eDNA, they don't really need to be, because they're going to be spending a lot of time still identifying insects and just having them sequenced.
Okay, we have some new questions here. Are samples able to be achieved for future sequencing?
It can be achieved. It could also be archived.
Yeah, sorry, archived.
Sorry, I got it. They can be. So I know that some, first let me recognize, looking at different names and pictures that are on here, we have people that are very familiar with this process and know a lot about it, more than I do in many respects. The DNA that we get, once we go ahead and extract it, we have roughly 100 microliters of DNA extract. We'll use a small amount of that, and then the rest of that can be frozen and archived for future sequencing. And what that means is that as we develop different primer sets to look at phytoplankton differently, or insects or fish, or then we start to go after different taxonomic groups, we can go back, resample those archives, and what happened in 2021 can be assessed in 2030. So all of that can happen. The process for archiving those and who's going to hold those in a repository, whether that will happen with a private firm like us, whether the state will do that, or some federal agency or quasi-agency like the Smithsonian, is something that still needs to be worked out.
Okay, and the next question is, are there any plans to quantify viral sequences?
That's a good question. Viruses are a pain in the butt because they're really diverse. Some of them are RNA, some of them are DNA, and there really aren't primers for viruses. And because their genomes are relatively small and there's very little that's conserved across viruses. So to do that, you have to do some pretty major heavy duty sequencing to just get everything. And then you pick out the viral genomes too. So the good news is those archives, those DNA archives that we have, for the most part, can be used to go after viruses at a later point. Other pathogens that are there, like fungal pathogens, bacterial pathogens, are also going to be, their DNA will be extracted, and we can go after those too. Viruses are probably the hardest out of all of them.
Okay, the next question is, if we are trying to determine the algal species of macroinvertebrates in our stream, how would a sample be collected or homogenized to fit through a filter?
So if we have that's a good question. So what we have focused on has at first just been, how do we suck up water, push it through a filter, and take it from there? As we know that there's other traditional techniques that have developed with other goals that are physical sampling. And so kicknuts, where we get a bag of bugs, quote unquote, trademark hashtag, and as well as paraphyton, which is a different indicator, it's a different index that essentially we have to figure out how to homogenize those. As far as bags of bugs are concerned, we essentially just put it through a blender, and then we take some of the blendate and sequence that. For paraphyton, it's kind of the same approach. Any paraphyton that would be put into a small jar, for example, then gets homogenized in a couple of different ways. So we can homogenize physical specimens too, and take that homogenate and sequence it just like we would the filter.
Okay, and it looks like we have, excuse me, a request from Peter here. Susie, can you tell us about your metabarcoding project and how you think results will stack up? How many samples are you collecting?
Sure. So as Peter mentioned, we have our algal DNA metabarcoding project that's been going on for the past almost 5 years, where every field season, we've been asking a bunch of our collaborators who are collecting stream samples to collect alongside their algal morphology sample and algal DNA sample. And so now we've been going through the process of extracting and sequencing those couple hundred samples. So we have over 1,000 samples in hand right now. We've sequenced about half of them, and they are all being used to see how well we can calculate algal stream condition index scores using DNA-based approaches. And what I'm really excited about with Peter's project is because you'll be collecting water samples and sequencing them and getting phytoplankton sequence data, seeing who's in the water column and how that relates to who's in the benthic community that we've been sequencing through our algal barcoding metabarcoding project. So I think there'll just be really great opportunities to compare both who's in the water column, who we're picking up maybe in our fish eDNA signatures, and how that relates to the biological condition information that we're getting from our ASCII molecular data. Like Peter was saying, we've encountered a lot of the same types of questions through the process of developing the algal DNA program, such as how do you handle a DNA reference database that sometimes has gaps in it? How does that, what are the implications for how you're analyzing your sample if you know you might be missing some certain taxa? How can we begin to fill those gaps? How do you handle abundance data? What are our favorite DNA barcode regions to be using to get the best representation of the algal community in our sample? So lots of similar types of questions that we've been asking for our algal data set that now you're going to be asking for your water eDNA, both your fish and your benthic microinvertebrates and all of your 23S phytoplankton data also. So I think there's going to be really great opportunities to compare the information that we've gotten from those two different projects. So I'm excited to see it all come together.
And then on that note, I'm Nathan Mackin with Department of Fish and Wildlife. We've been some of the field crews that have been collecting those eDNA samples for Susie. And this year I received, I believe it was 18 kits or so. I'm not sure if that was the right number, Peter. We've been collecting water column eDNA alongside samples that we're going to send to Susie. So hopefully that'll, she'll be able to see that comparison soon. I just wanted to add, or I was going to type it in the chat if I didn't say it here, that whatever gaps you're finding between physical taxonomy and eDNA, let me know. Maybe we can go through and see where we do have the physical taxonomy. And with whatever kits I get this next year or not, I can try to target certain locations and maybe pick different reference sites according to where the gaps are to try to clear those up sooner than later.
That's awesome. And that's one of the things that we've been looking for in some of our data sets when we know that we have an algae that we pick up a ton in all of our microscopy samples, but we know we're missing a reference sequence for it. So we can't necessarily pick it up in our metabarcoding samples, identifying what streams or regions would we want to try and do some more aggressive sampling to hopefully generate a sample that we could do the sequencing for the reference metabarcode sequence ourselves. So that's awesome. I think comparing that type of data is going to be really great.
Yeah, if there's anything special you're looking for, just reach out to me and I'll
Thanks so
see
much.
what I can do. Yeah.
Perfect.
Or the week after, I can't remember, but we're meeting shortly to talk about next year, and I'm super excited about it. This sounds great.
Hopefully our programs will go back to normal where we get to cover the whole state. The last 2 years because of COVID have been pretty wacky. We've been kind of had like a moving target the whole year. But I'm expecting next year to be a more normalized year too. And
I know you guys, Nathan, are typically you don't go to sites annually, but you are now. Is that correct? You'll be visiting the same site over and over?
Mm-hmm.
How many sites approximately?
So it would be, well, we have multiple projects, but within the RCMP, we've had, I think it was about 8 or 9 sites that we'd visit repeatedly. So some of those sites will get like up to 5 or 6 visits. Some of them are only at 3. Depends on, some of our other, what other shoes got thrown into the cogs. The fires have kind of messed stuff up for us as well. Like we had sites that we'd sample a couple times and then they caught on fire, stuff like that.
Wow.
But in general, I've been only using the kits 1 per site since I figured the greatest number of comparisons between Susie's and Joseph's samples would be the best idea. Unless somebody has a different idea. I mean, if you'd rather do multiple samples across time, we could do something like that in the future too.
I was going to kind of throw that out there. And it's been because I think it's great that you used 1 per site because we have a limited number of kits. But the future is looking bright. So what would be the benefits of dupes or taking triplicates while we're out there? And this is for Joe, Susie, anybody that wants to field it.
I think, Susie, Nathan, if it's okay, I'll start. We've essentially there's effort response curves that look at species accumulation. When we sample 100 milliliters of water, you're not necessarily going to get all the DNA in that one filter. And even then, we only take a portion of what we extract from that filter. And so we actually run it through the lab process 6 different times each filter. As we get more and more filters, you're going to pick up rare and rare taxa on there. So to have we don't have a standard for how many samples that that are best to take for a particular type of site. So some sites do we just need 1? For others, is it going to take 3 or 4? And if we start to put essentially diversity effort curves together, richness effort curves together, we will get a better idea of that. More than likely for something like phytoplankton, 1 should be pretty good. For rarer stuff like fish, or if we start to go to hard stuff like amphibians or other mammals that are associated with water, we'll probably need more and more. So there's some basic research that needs to be done on that. It's really, for the most part, it's a little bit of effort and it's a budgetary question more than it is a scientific question. The science is pretty easy to do once we get the samples.
I would definitely echo what Joe is saying. If there is a site in particular where you maybe have an organism is present but in low abundance, doing something like taking multiple replicates, of course, increases your chances of catching that organism. And it would be nice to have some preliminary information. And like you said, it can really vary by your target taxa and your actual situational environment, how many replicates would be optimal for detecting those organisms. But having at least a couple of sites where you're taking those replicates are always informative. And so I think that's awesome. The other thing I was thinking that would be really neat at some point to do is if you had a known occurrence, maybe somewhere upstream of where you're sampling, and we could do some sort of transect-based sampling where you're detecting increasing distances downstream. That's, again, the type of information that we're trying to have more and more of for all sorts of different environments across the state to see where put some boundaries on those propagation estimates of how far that eDNA is transiting from its original source organism. So any of those types of questions would be really cool. And I'm excited to see what we can do.
Grab at Stanton Creek and Stony Creek, 3 different samples, quarter-mile downstream each. So we'll see how those turn out. That was more of a, oh, I got to get rid of these kids question instead of a planned question, unfortunately. But the COVID thing really knocked us for a loop. But I'll be excited to see how those differ and turn out. Do we have any more questions?
Nope, we're all caught up.
All right. If we have no more questions, I guess this is where we'll leave it. I want to thank everybody for your help. I'm so excited about doing this and getting it to grow. And something, Joe and I were in a meeting a while back, and he was explaining, his vision, and he said something about how this is what keeps me up at night. And I chuckled at first because I'd never really heard that expression, but I completely understood it when I was lying in bed thinking about, how we can improve our project. So I apologize for chuckling about that, Joe, because I feel it. I'm super excited.
I feel bad that it's caused you some insomnia. That's nothing I would wish upon anybody. Yeah, that's all.
It's the good kind. So I'm super stoked about it. I just hope we get a lot of participation in this thing. I want to get it to spread all over the state, and really get, some different elevations, real different geographic location stuff, and just see where we can go with this. I'm very excited to be a part of this new technology. It's going to be great.
Mm-hmm. And if I can just echo for a second too, like I never really talked about my personal experience on this, but it's fun to do this. I mean, taking a sample takes about 5-10 minutes, and then a little bit later you get data back, and it's just fascinating to see what was in that stream. It's not as much fun necessarily as going shocking or doing staining, but at the same time, it's a different way to interact with the environment. And we always will pick up things that we just never expected were there because we just didn't know. And that's not just fish, but also, we'll get bycatch for different mammals that have passed through, for example, and also different amphibians. So it's a really fun way to just learn more about the biodiversity. I still, every time we get new data in, I'm looking at where these samples are coming from, and and whether that's finding sharks in the Chesapeake Bay or dolphins elsewhere. And even small things that we don't really think about as far as chubs and shiners and things like that. It's you'll spend time Googling up species names, that's for sure. Thanks, Peter.
Thank you, Joe. I'm just reading that, making sure we don't have a question. Okay. Looks like we're all good. Thank you all for joining up. Please sign up if you can, sign up for the email list. Let's get those kits out there and start sampling.
Thanks, everybody. Thank you, Peter. Good to see you, Susie.
Thanks,
Take
Peter.
care, guys.
Bye.
Thanks, guys. Hey, Peter, this is Eric with Clean Water Team.
Hey, Eric.
Hey, a lot of stuff was being discussed. I didn't want to interfere the flow. Do you got maybe 3 minutes to chat?
Absolutely, man.
Yeah, as your questions get more formulated,